Cellular homeostasis relies upon the careful regulation of the movement of molecules and ions across cellular membranes. For polar substrates, membrane translocation is carried out by membrane transport proteins. There are two main classes of membrane transport proteins: passive membrane transport proteins, which facilitate movement of material across the membrane in the same direction as the concentration gradient and do not require an external energy source, and active membrane transport proteins, which often use an energy source, such as ATP hydrolysis, to translocate molecules or ions against a concentration gradient. The potassium channel protein is an example of a passive membrane transport protein that translocates potassium ions across membranes in both bacteria and eukaryotes. The potassium channel shows a remarkable selectivity for potassium ions, allowing only one sodium ion through for every 10,000 potassium ions despite close similarities in ionic size and charge. Structural analyses of the tetrameric potassium channel show that this selectivity is due to a narrow opening, called the selectivity channel, within the interior of the potassium channel protein complex. Researchers propose that both potassium and sodium ions can initially enter the channel in a solvated state, that is, surrounded by water molecules. The interaction of potassium ions with oxygen atoms on amino acids in the selectivity channel allows them to desolvate or shed their water molecule associations and pass through the channel. Sodium ions, being slightly smaller than potassium ions, cannot make the same interaction as potassium ions. As a result, it is energetically unfavorable for sodium ions to desolvate, and their large solvated size keeps sodium ions from moving through the channel. The sarcoendoplasmic reticulum calcium ATPase, or SERCA, is an example of an active membrane transport protein. SERCA is abundant in muscle cells, where it transports calcium ions from the cytoplasm into the lumen of the sarcoplasmic reticulum to promote muscle relaxation. SERCA is inactive when associated with a second membrane protein called phospholambin. Muscle relaxation is initiated when phospholambin is phosphorylated by regulatory kinases, causing it to dissociate from SERCA. SERCA is thought to undergo a four-step process to transport calcium ions. First, 2 or 3 hydrogen ions are exchanged for 2 calcium ions on the cytoplasmic side of the membrane. Next, aspartate 351 of one of the SERCA subunits is catalytically phosphorylated by ATP to form an aspartyl phosphate moiety, which results in a conformational change that allows calcium ions to exit the binding pocket and enter the lumen, and also allows hydrogen ions to enter. In the third step, the binding of ATP induces a conformational change that seals the opening and traps the hydrogen ions in the binding pocket. In the final step, dephosphorylation of aspartate 351 returns the protein to its original conformation, allowing the hydrogen ions to be exchanged for calcium on the cytoplasmic side. The potassium transporter and SERCA serve as illustrative examples of passive and active membrane transport and the interdependence of protein structure and function.